Hi, today we're going to learn about gene regulation in E. coli through the Lac Operon. So why is it called the Lac Operon? This is because the Lac Operon controls lactose metabolism in E. coli. Okay, so to understand the Lac Operon in simple terms, pretend you're walking down the road on your way to work and there's a huge boulder in the way. Obviously you can't move, but then pretend there's an imaginary force that gets rid of the boulder, and then you can make your way down to work and get your job done. Simply, that's how the Lac Operon works. So as I said, the goal of the Lac Operon is to control the translation and transcription of the genes that allow for lactose metabolism. Those are the three main structural genes: Z, Y, N. And these are your structural genes. Z is the gene for the enzyme known as beta-galactosidase, which basically hydrolyzes the lactose, while Y is permease, which permits the entry of the lactose into the cell, and then transacetylase basically transfers the acetylates during the process of lactose metabolism. Okay, so pretend there were a high concentration of lactose. Then these genes, the transcription of these genes would need to be turned on, right? Because you would need those enzymes to break down the lactose. But if there was no lactose present in the cell's environment, then why would you transcribe and translate those genes? You wouldn't need to do that because there is no lactose, so what are you going to do with those enzymes? That's why the operon needs to be turned on and turned off. So now we're going to go into the details of how exactly the operon is turned off and how exactly it's turned off. So as I said before, the goal of the Lac Operon is to control the transcription and translation of these three genes over here. So now if you see, there is no lactose present, so you would need this to be turned off. So basically, as you know, the promoter region is where, we'll think of it as P, but it's the promoter region. The promoter region is where RNA polymerase binds to start transcription. But how would we turn this off? How would we prevent RNA polymerase from actually transcribing these genes? Well, there would need to be an obstruction in its way, similar to how there was a boulder blocking you from getting to work. This is the operator region, and this is where the repressor would bind. The repressor basically represses RNA polymerase. It prevents it from transcribing these genes. Now pretend there was a high concentration of lactose. Then you would need these three structural genes to be transcribed, right? So what would happen then? Basically, the Lac Operon is an inducible system, meaning the inducer is what can activate the transcription and translation of these genes. So what would happen is that this lactose, it would take an allosteric form. Allosteric basically means that it's like an isomer. So the chemical formula is the same, but the structure is different. So allolactose, which is an allosteric form of lactose, would actually bind to the repressor. And this would only be possible if lactose was there in the environment, obviously. So then upon binding to the repressor, the shape of the repressor would change. And if its shape changes, then it can no longer bind to the operator region, and it would move away, and then transcription and translation of those genes would continue because RNA polymerase could continue moving. Now, you may be wondering, where does this repressor comes from? Well, that's where I comes in. I is not a part of the actual operon, and I'll tell you why right now. So the definition of an operon is important to this lesson, obviously. An operon is a cluster of genes with a single promoter region. These genes have a single promoter region, but I has its own promoter and its own terminator. that's why I is not part of the actual Lac Operon. The actual Lac Operon is only POZYA. So now if we were to address where does the repressor comes from, the I gene is what it actually codes for the repressor. Okay, so far we have learned that POZYA is the actual operon because it has a single promoter region, and that I is separate from it because I has its own promoter and terminator. Now, a way you can remember that I actually codes for the repressor is that think of it as inhibitor, because basically what the repressor does is that it inhibits the activity of RNA polymerase. Okay, so far we took into consideration what would happen if there was a concentration of lactose in the environment. Now, what would happen if there was lactose and glucose? Well, since glucose is always the preferred source of energy, the lactose would not be broken down, transcription of these genes would not occur properly because glucose is the preferred source of energy. There is a scientific mechanism behind that, which we will discuss right now. So first, what we're going to do, I'm just going to extend this promoter region because there is another factor that comes into play that actually binds to the promoter region. And then we have I over here. Okay, so the other thing that binds to the promoter region, which I'm going to draw over here, is CAP. CAP is basically the catabolite activator protein, and then CAP binds to CAMP, and they form a CAP-CAMP complex. So what CAMP is actually important to understanding why lactose gene transcription would not occur. This is because CAMP is basically cyclic adenosine monophosphate, and by the name you could tell that it's basically a broken down version of ATP. So to break down glucose during glycolysis, Krebs cycle, all those steps, you're going to need amounts of ATP. That means that there will be a low amount of CAMP. And if there's a low amount of CAMP, CAMP won't be able to properly bind to CAP. And then the CAP-CAMP complex, what it does is that it's actually supposed to bind over here. So it binds to the promoter region as well as to the RNA polymerase. And basically the greater the binding of the CAP-CAMP complex, the greater the transcription rate. So it enhances transcription. But if there's a low level of CAMP, because the ATP is actually being used to break down glucose during glycolysis, Krebs cycle, all those steps, there will be a low level of CAMP. And if there's a low level of CAMP, that means the CAP-CAMP complex won't form properly. And if the CAP-CAMP complex doesn't form properly, it won't properly bind to the RNA polymerase. And if it doesn't bind to the RNA polymerase, then transcription won't occur properly. So basically what's happening is that since the cell is trying to allocate all of its ATP to breaking down glucose, then there's going to be a low amount of CAMP, and hence transcription of these genes that help metabolize lactose won't occur properly. So today we basically covered the Lac Operon and gene regulation in E. coli. So if you guys have any questions, you can let me know in the comments, and the next video in this series will be based on the tryptophan operon, which is a different type of operon since it's a repressible system. I'll go into more details on that in the next video.